I'm Dr. Connie Fung, and I'm a postdoctoral research fellow in the lab of Dr. Michael Howitt at Stanford University. More than 1 billion people worldwide are infected with parasitic worms, or helminths. That amounts to about 1 in 8 people globally who harbor these organisms inside their intestines. That is a staggering number. Now, you might be surprised at this statistic. In a sense, worm infections are a forgotten disease in the developed world. Thanks to widespread treatment efforts, education, and improved sanitation, helminth infections have become pretty uncommon here. However, parasitic worm infections are still very much an everyday reality for those who live in regions of extreme poverty. They are some of the most neglected diseases in healthcare systems and scientific research. This is mostly due to a lack of awareness. Helminth infections are rarely found in big cities. Instead, they disproportionately affect those living in poor rural communities. Helminth infections are also generally not fatal. They do not kill to the same extent as more well-known infectious diseases such as HIV, AIDS, or malaria do. However, they still endanger human health in debilitating ways. Worm infections often cause chronic illness that leads to malnourishment, stunted growth, impaired cognition, and long-term disabilities. Young children, in particular, tend to harbor higher worm burdens in their guts and suffer the most from these debilitating outcomes. These negative effects on child health can hamper their ability to go to school and perform well academically. This can also lead to reduced productivity at work in the future and trap these individuals in a continuous cycle of infection and poverty. Since 2001, the World Health Organization has advocated for the control of helminth infections in endemic regions. They did so through a mass drug administration program where more than 700 million people who live in high-risk communities are given deworming medications every year. Now, despite these efforts, controlling worm infections still remains a huge public health challenge. Treated individuals in endemic areas often get reinfected over and over again. This is because of a lack of adequate sanitation infrastructure. The environment is continuously being contaminated with worm eggs and larvae that were shed by infected individuals. Plus, by constantly exposing worms to anti-helminthic medications, drug-resistant parasites can emerge. We need more effective therapies to supplement current deworming efforts. Examples include drugs that can enhance host clearance responses rather than directly targeting the worms, or vaccines that can prevent infection altogether. I'm interested in studying how the host detects and responds to intestinal worm infections. What aspects of our biology can we target to enhance clearance of these parasites? Investigating these questions is a crucial first step in the path to developing novel therapies for these debilitating diseases. I was first introduced to parasites in my undergraduate parasitology class. At the time, I had no idea what they were, and I was really surprised to learn not only about their unusual biology, but also the massive impact that these infections had on the poorest populations in the world. Inspired by this newfound knowledge, I began doing research on parasitology as an undergrad. Now, as a child, I've always loved asking questions and learning new things. So it's not surprising then that I was super excited to make new discoveries about parasites and also really grew to love research. However, I was also hoping that as I further develop as a scientist, my work can eventually contribute to helping people who are vulnerable to these diseases. This experience inspired me to go to graduate school to continue my science training. For my PhD, I studied pathogens that infect the GI tract. Combined with my previous research experience with parasites, this led me to where I currently am as a postdoctoral researcher, studying parasitic worms that infect the intestine and how our bodies respond to them. Our intestines are lined with a single layer of epithelial cells with many different subtypes that have their own unique functions. Together, these epithelial cells form a physical barrier that protects us from harmful things in the environment, like pathogens. Epithelial cells can sense molecules that are produced by different types of pathogens, and then they can alert the immune system that there is an infection. In the case of intestinal worm infections, there is one crucial epithelial cell type that initiates an anti-parasite response within the host. These specialized cells are known as tuft cells, and they are named as such because they have a tiny tuft of hair-like projections that reaches out into the intestinal space. These projections are studded with taste receptors, proteins that we would normally find on the taste cells in our tongue. These receptors help us taste sweet, bitter, and umami flavors from the food that we eat. So, what are they doing on tuft cells in the gut? Well, given their location, it's likely that these intestinal taste receptors aren't contributing to our sense of taste. As it turns out, these receptors can actually taste molecules that are secreted by parasites, allowing tuft cells to detect a worm infection. Upon tasting the presence of worms, tuft cells release signaling molecules to alert nearby immune cells that there are parasites in the gut. This enables the immune system to mount an anti-parasite response to clear these invaders from our bodies. My research hopes to uncover novel signaling molecules released by tuft cells and how tuft cells use these messengers to communicate with various cell types in our bodies and direct them to clear parasites from the gut. I'm hopeful that my work can contribute in the global fight to control worm infections, to reduce morbidity among the most vulnerable populations, especially young children, and to eventually eradicate these diseases.